Hello everyone, and welcome to the International Museum of the Human Cloning. Today we will teach you a little bit about the history of human cloning. In the tour, we will visit 4 historians who will tell you the story, and well, I hope you like it.
I will tell you the story of human cloning before the 60s, because all of this led to the scientists and policymakers to take the prospect seriously. In 1885, German scientist Hans Adolf Eduard Drich began his research on reproduction, where the first study of cloning took place. Until 1902, Walter Sutton proves chromosomes hold genetic information.
For me, that year was the biggest step, discovering the genetic information. Wow!
Yep. Then in 1938, a German scientist called Hans Spemann proposed a fantastical experiment to transfer one cell nucleus into an egg without a nucleus, which is basic method that would eventually be used in cloning. Could you imagine it? And finally, in 1953, Watson and Crick find the structure of DNA. What are your thoughts on the subject? What do you think will happen next?
Let's continue with the tour, and now between the 60s and 12s, we have Andrea. Hi Andrea.
Hi. As it was said before, scientists and policymakers began to take the prospect seriously in the 60s. It all started in 1963 when Mr. Haldane introduced the idea of human cloning, for which he used the terms clone and cloning. In 1966, the Nobel Prize-winning geneticist...
Wait, Nobel Prize-winning geneticist? Wow!
Yes, the Nobel Prize-winning geneticist Joshua Lederberg advocated cloning and genetic engineering in an article in the American Naturalist. And again, the following year, in the Washington Post, he sparked a debate with the conservative bioethicist Leon Kass, who wrote at the time that the programmed reproduction of men will in fact dehumanize him.
Yeah, I think that for those times it would be really hard and bad to think in cloning a person without having investigated about it.
Well, that's why in 1987, a document entitled Introduction on Respect for Human Life and Its Origin and on the Dignity of Procreation, published by the Congregation for the Doctrine of Faith, Roman Catholics, were told that cloning is...
Ooh, here comes the best!
Finally, in 1996, the cloning of a sheep known as Dolly by somatic cell nuclear transfer, SCNT, made the idea of human cloning become a hot debate topic. This led a lot of nations to outlaw it, while a few scientists promised to make a clone within the next few years. Later on 1998, the first hybrid human clone was created by Advanced Cell Technology, which developed into an embryo, but it was destroyed 12 days later. Do you agree with the Roman Catholics' perception of cloning? Is it wrong and goes against our morals? Well, there is a lot more for this story.
Thanks, Andrea. Now let's continue with the next historian who will tell you the story between 2004 and 2008. With you, Adrian.
Hello guys, how are you doing? In 2004 and 2005, Juan Wo Suk, a professor at Seoul National University, claimed to have created 11 different patient-specific stem cell lines, and then the first clone of a domestic dog was created. Sadly, in 2006, Science retracted both articles of Juan Wo Suk on clear evidence that much of this data from the experiment was fabricated. In January 2008, Dr. Andrew French and Samuel Wood of the biotechnology company Stemagen announced that they successfully created the first mature human embryos. Each embryo was created by taking a nucleus from a skin cell and inserting it into a human egg from which the nucleus had been removed. The embryos were developed only to the blastocyst stage, at which point they were studied in processes that destroyed them. What do you think had happened after this? Or do you think what he did was right?
Really interesting, Adrian. And finally, we will meet the last historian who will tell us the story from 2011 to the present.
In 2011, scientists at the New York Stem Cell Foundation announced that they had succeeded in generating embryonic stem cell lines, but their process was not useful. In 2013, a group of scientists led by Shukrat Mitalipop published the first report of embryonic stem cells created using SCNT. The researchers developed a protocol for using SCNT in human cells, which differed slightly from the one used in other organisms. A year later, a team led by Robert Lanza at Advanced Cell Technology reported that they had replicated Metalipop's result and further demonstrated the effectiveness by cloning adult cells using SCNT. Finally, in 2018, the first successful cloning of primates using SCNT was reported with the birth of two live female clones, crab-eating macaques named Song Song and Hua Hua.
Well, here's where the tour finishes. I hope you've liked it, but tell us what do you think about this? Do you think cloning is good? Or what do you think about that could happen in the future? Come back soon and thanks for watching us.